reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediate ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complex within the photosystem 1 and the photosystem 2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigment molecules bound to protein. Each photosystem has all the pigment except one molecule of chlorophyll A, forming a light harvesting system, also called antenna. Figure 13.4. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystem. In photosystem 1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called photosystem 700. Sorry. While in photosystem second, it has absorption maxima at 680 nanometer and is called P680. 13.6. The electron transport in photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electron to become excited and jump into an orbit further from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of cytochromes that is given in figure 13.5. This movement of electron is downhill in terms of an oxidation, reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on the pigments of photosystem 1. Simultaneously, electrons in the reaction center of photosystem 1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy rich NADP+. The addition of these electrons reduces NAD plus to NADPH plus H plus. This whole scheme of transfer of electron starting from photosystem second uphill to the acceptor, down the electron transport chain to photosystem 1, excitation of electrons transferred to another acceptor, and finally downhill to NADP plus, reducing it to NADPH plus H plus, is called the Z scheme due to its characteristic shapes. Figure 13.5. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. 13.6.1. Splitting of water. You would then ask, how does photosystem 2 supply electron continuously? The electrons that were moved from photosystem second must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the photosystem 2. Water is split into 2H plus O 2H plus and electron. This create oxygen. 2H plus and of oxygen and electrons. These create oxygen, one of the net product of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. We need to emphasize here that the water splitting complex is associated with the photosystem 2, which itself is physically located on the inner side of the membrane of the thylakoid. Then, where are the protons and O2 formed likely to be released in the lumen or on the outer side of the membrane? 13.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substance and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first photosystem second and then the photosystem one, a process called non-cyclic photophosphorylation occur. The two photosystems are connected through an electron transport chain, is as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by these kind of electron flow that is given in figure 13.5. When only photosystem 1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening is in the stroma lamella. While the membrane or lamellae of the grana have both photosystem 1 and photosystem 2, the stroma lamellae membranes lack photosystem 2 as well as NADP reductase enzyme. The excited electron does not pass on to NADP plus but is cycled back to the photosystem 1 complex through the electron transport chain that is given in figure 13.6. The cyclic flows hence result only in the synthesis of ATP but not of NADPH plus H plus. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation. 13.63 6.3 Chemio Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time these are the membranes of thylakoid. There is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria. When electron move through the ETS, let us understand what caused the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electron and their transport to determine the steps that cause a proton gradient to develop. Since splitting of the water molecule takes place on the inner side of the membrane, the proton or hydrogen ion that are produced by the splitting of water accumulate within the lumen of the thylakoid B, and as the electron move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule remove a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane along with electron that come from the acceptor of electron of photosystem 1. Protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number while in the lumen there is accumulation of proton. This creates a proton gradient across the thylakoid membrane as well as measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of proton across the membrane to the stroma through the transmembrane channel of the CFO of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of proton across the membrane. The, the other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provide enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which make the enzyme synthesize several molecules of energy packed ATP. Chemiosmosis require a membrane, a proton pump, a proton gradient, and ATP synthase energy is used to pump proton across a membrane to create a gradient or a high concentration of proton within the thylakoid lumen. ATP synthase has a channel that allows diffusion of proton back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyze the formation of ATP along with the NADPH produced by the movement of electron. The ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugar. 13.7 Where are the ATP and NADPH used? We learn that the product of light reaction are ATP, NADPH and O2. Of these, O2 diffuses out of the chloroplast while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the product of the light reaction, that is ATP and NADPH. Besides CO2 and H2O, you may wonder how this could be verified. It is simple. Immediately after light become unavailable, the biosynthetic process continues for some time and then stop. If then light is made available, the synthesis start again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourselves. Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O or sugar. It was of interest to scientists to find out how this reaction proceeded, or rather, what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive carbon 14 by him in algal photosynthesis study led to the discovery that the first CO2 fixation product was a 3 carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it was called Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation or whether any other product was formed in the other plant. Experiment conducted over a wide range of plants led to the discovery of another another group of plant where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: those plant in which the first product of CO2 fixation is C3 acid PGA, that is C3 pathway, and those in which the first product was a C4 OAA, that is the C4 pathway. These two groups of plants showed the other associated characteristic that we will discuss later. 13.71 The primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after accepting CO2, would have 3 carbons of PGA? The study were unexpectedly so that the acceptor molecule was of a 5 carbon ketose sugar ribose biphosphate RUBP. Did any of you think of this possibility? Do not worry. The scientists also took a long time with and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2 carbon compound. They spent many years trying to identify a 2 carbon compound before they discovered the 5 carbon RUBP. 13.72 The Calvin cycle. Calvin and his co-worker then worked out the whole pathway and showed that the pathway operated in cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates, where the sugar is synthesized. Let us at the outset understand very clearly that the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathway that is given in figure 13.8. For case of understanding, the Calvin cycle can be described under 3 stages: carboxylation, reduction, and regeneration. First, carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which result in the formation of two molecules of 3-phosphoglyceric acid. Since these enzymes also have an oxygenation activity, it would be the more correct to call it RUBP carboxylase oxygenase or Rubisco. 2. Reduction. These are a series of reactions that lead to the formation of glucose. The steps involve utilization of two molecules of ATP for phosphorylation and two of NADPH for reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of 1 molecule of glucose from the pathway. Third, regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration step require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Calvin pathway. It might help you to understand all of this if we look at what goes in and what comes out of the Calvin cycle. In that is given in the table readout. 13.8 The C4 pathway. Plants that are adapted to dry tropical region have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way are they different from C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperature. They show a response to high light intensities. They lack a process called photorespiration and have a greater productivity of biomass. Let us understand these one by one. Study vertical section of leaf, one of a C3 plant and the other of a C4 plant. Do you notice the differences? Do both have the same types of mesophyll? Do they have similar cells around the vascular bundle set? The particularly large cells around the vascular bundle of the C4 anatomy are said to have a Cranch anatomy. Cranch means breadth and is a reflection of the arrangement of cell. The bundle form the bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick walls impervious to gaseous exchange, and no intercellular space. You may like to cut a section of the leaves of C4 plants, that is maize or sorghum, to observe the Cranch anatomy and the distribution of mesophyll cell. It would be interesting for you to collect the leaves of diverse species of plants around you and cut vertical section of the leaf. Observe under the microscope. Look for the bundle sheath around the vascular bundle. The presence of the bundle sheath would help you identify the C4 plants. Now study the pathway shown in figure 13.9. This pathway, that has been named the Hatch and Slack pathway, is again a cyclic process. Let us study the pathway by listening the steps. The primary CO2 acceptor is 3 carbon molecule phosphoenol pyruvate PEP and is present in the mesophyll cell. The enzyme responsible for this fixation is PEP carboxylase or pepcase. It is important to register that the mesophyll cell, like Rubisco enzyme, the C4 acid OAA is formed in the mesophyll cell. It then forms other 4 carbon compound like malic acid or aspartic acid in the mesophyll cell itself, which are transported to the bundle cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and a 3 carbon molecule. The 3 carbon molecule is transported back to the mesophyll where it converted into PP again, thus completing the cycle. The CO2 released in the bundle cyst cells enters the C3 or the Calvin pathway, a pathway common to all plants. The bundle cyst cells are rich in an enzyme ribulose biophosphate carboxylase oxygenase Rubisco, but lack pepcase, thus the basic pathway that result in the formation of the sugar. The Calvin pathway is common to the C3 and C4 plants. Did you note that the Calvin pathway occur in all the mesophyll cell of the C3 plant? In the C4 plants, it does not take place in the mesophyll cell but does so only in the bundle sheath cells. 13.9. Photorespiration. Let us try and understand one more process that creates an important differences between C3 and C4 plants. Photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Calvin pathway, the first CO2 fixation step. This is the reaction where RUBP combines with the CO2 to form two molecules of 3-phosphoglyceric acid, that is catalyzed by Rubisco. Rubisco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? Rubisco has a much greater affinity for CO2 when the CO2 versus O2 is nearly equal. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of O2 and CO2 that determine which of the two will bind to the enzyme. In C3 plants, some O2 does bind to Rubisco and hence CO2 fixation is decreased. Hence the RUBP, instead of being converted to molecules of phosphoglyceric acid, binds with the O2 to form one molecule of phosphoglycerate and phosphoglycolate, that is two carbon structure, in a pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugar nor of ATP. Rather, it results in the release of CO2 with the utilization of ATP. ATP in the photorespiratory pathway. There is no synthesis of ATP or an ADPH. The biological function of photorespiration is not known yet. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increase the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle itself to release CO2. This result is increasing the intracellular concentration of CO2. In turn, this ensures that the Rubisco function as a carboxylase, minimizing the oxygenase activity. Not now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperature. Based on the above discussion, can you compare plants showing the C3 and the C4 pathway? Use the table format given and fill in the information. 13.10 Factors affecting photosynthesis. An understanding of the factors that affect the photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal plant and external. The plant factor include the number, size, age, and orientation of leaves, mesophyll cells, and chloroplast, and internal CO2 concentration and the amount of chlorophyll. The plant or internal factor are dependent on the genetic predisposition and the growth of the plant. The external factor would include the availability of sunlight, temperature, CO2 concentration, and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, though several factors interact and simultaneously affect photosynthesis or CO2 fixation, usually one factor is the major cause or is the one that limit the rate. Hence, at any point the rate will be determined by the factor available at suboptimal level. When several factors affect any biochemical process, Blackman's 1905 law of limiting factor come into effect. This states the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affect the process of its quantity is changed. For example, despite the presence of a green leaf or an optimal light and CO2 condition, the plant may not photosynthesize if the temperature is very low. This leaf, if given the optimal temperature, will start photosynthesizing. 13.10.1 Light. We need to distinguish between light quality, light intensity, and the duration of exposure to light. While discussing light as a factor that affects photosynthesis, there is a linear relationship between the incident light and CO2 fixation rate at low light intensities. At higher light intensities, gradually the rate does not show a further increase as other factors become limiting. What is interesting to note is that light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. 13.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rate. Beyond this, the level can become damaging over longer periods. The C3 and C4 plant respond differently to CO2 concentration at low light condition. Neither group respond to high CO2 condition at high light intensities. Both C3 and C4 plants show increase in the rate of photosynthesis. What is important to note is that the C4 plants show saturation at about 360 micro per liter, while C3 respond to increased CO2 concentration and saturation is only beyond 450 micro per liter. Thus, current availability of CO2 level is limiting to the C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yield. 13.10.3 Temperature. The dark reaction being enzymatic are temperature controlled, though the light reaction are also temperature sensitive. They are affected to a much lesser extent. The C4 plants respond to higher temperature and so higher rate of photosynthesis, while C3 plants have much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have higher temperature optimum than the plants adapted to temperate climate. 13.10.4 Water. Even though water is one of the reactant in the light reaction, the effect of water as a factor is more through its effect on the plant rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Beside water, stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Summary. Green plants makes their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by leaves through the stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cell have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are site for the light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages: the light reaction and the carbon fixing reaction. In the light reaction, the light energy is absorbed by the pigment present in the antenna and funneled to a special chlorophyll A molecule called reaction center chlorophylls. There are two photosystem 1 and photosystem 2. Photosystem 1 has a 700 nanometer absorbing chlorophyll A photosystem 700 molecule at its reaction center, while photosystem 2 has a photosystem 680 reaction center that absorb red light at 680 nanometer. After absorbing light, electrons are excited and transferred through photosystem 2 and photosystem 1 and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to movement through the F0 part of the ATP is enzyme release enough energy for synthesis of ATP. Splitting of water molecule is associated with the photosystem 2, resulting in the release of O2 protons and transfer of electron to photosystem 2. In the carbon fixation cycle, CO2 is added by the enzyme Rubisco to a 5 carbon compound RUBP that is converted to 2 molecule of 3 carbon PGA. This is then converted to sugar by the Calvin cycle, and the RUBP is regenerated. During this process. ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plants. Photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4 carbon compound. In the bundle cells, the Calvin pathway is carried out for the synthesis of carbohydrates. Thank you friends for listening this audio. If you want any kind of, and please post your comment if you want any kind of improvement. So मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय, एंड जस्ट स्टे ट्यून्ड विद बायोलॉजी क्रिटिक एंड एंजॉय द नॉलेज.